Hi, so I'm Ryan Poplin, and so for the next chunk of time, we get to talk about my favorite topic in the world, which is quality scores. And so in this little session, I'm going to try to convince you, first of all, that quality scores are incredibly important. They're the most important concept in next-generation sequencing. And then next, I'm going to try to convince you that the quality scores, they're very important, but as they come out of the machine, they're incredibly inaccurate. Sequencing vendors do not do a very good job of estimating these confidences. And I'm going to show you that this program called the Base Quality Score Recalibration can make those exquisitely accurate. So here we go. Here's where we are in our little pipeline. So we've talked about all these things. Now we're down here. We've arrived at the third session. And really, the reason why these quality scores are just so incredibly important is that everything that comes after this, all of these various orange boxes, they're all just sophisticated statistical machinery. That's all it is. It's you give it evidence, and you're weighing the evidence in a Bayesian framework. So that single sentence sums up all the other tools in the GATK. And so because all you're doing is weighing evidence, that your estimation of the evidence is incredibly foundational, right? So let's take a look here. So the purpose is to estimate the evidence appropriately. So here we have an IGV screenshot, and we've shown you several IGV screenshots so far. Some have been very crystal clear and pristine. Some have been quite messy. I would say this one's sort of mediumly messy, right? So you come to this site, around here, and you want to decide, is this a real genetic variation? Is it just machine noise? What are all these green things? Are these green things real mismatches from the reference and it's genetic variation in the sample, or is it just machine noise, right? And so the way to do this is to come to these green things and to weigh the evidence. So I have a green base, and I'm 99% confident in that base. And I have this grayed out green base, and I'm only 20% confidence in that base, right? And so those quality scores or those estimates is what goes into your mutation calling procedure. So if all those estimates are wrong, then you're going to make the wrong decision in this case, right? If maybe you see here that that's 99% confident, but in fact it should be only 5% confident, you're going to perhaps call this as a real mutation when in fact you shouldn't. Right? So that's how this works. So the problem is the estimates as they come out of the machine, they're quite inaccurate, and worse, they're systematically biased, right? So what I'm showing you here is some example data, the original data as it came out of the machine, and recalibrated data after it's ran through this GATK procedure. And what I'm plotting on the one axis is the dinucleotide context. So you sequenced an A followed by another A, for example, in this bin, or you sequenced a G followed by another G. And on the other axis, I'm showing you the accuracy and the quality scores. So the machine said, of all those AAs, on average, it said they were, Q17, but in reality, they were Q whatever there is, right? And so what you see is there are crazy, crazy biases in these quality scores, right? So whenever you read an AA, you're overestimating the quality score, and whenever you read a G whatever, you're underestimating the quality score, right? And so these just has a crazy effect on your mutation spectrum when you call your samples, right? You're more likely to call A followed by an A than you would be whatever other thing, right? And furthermore, after you use this procedure, this bias is greatly reduced, right? So it's a flat profile along the dinucleotide context. You can measure that by the root mean squared error of these values. Here you have a 4 point something. Here you have a 0.2 something. So the bias is greatly reduced, right? Does that make sense? Oh, and the other thing I wanted to say is feel free at any time to jump in with questions. I like to have a more interactive sort of style, so jump in at any time. Okay.
Excuse me. On the left,
yeah, let's do it.
on the left panel, when you see this quantity is that you have two bases, right? The first base or the second base?
Okay. So the way this is defined is it's the second base. So you read the A followed by another A, and I'm showing you the estimate of the quality of that second A. Okay. All right. So you can define it any number of ways. What's beautiful about this software package is that you can write up a little Java class and write any covariate of error you'd like. If you care about the trinucleotide context instead of the dinucleotide context, it's very easy to investigate those sorts of things. you simply write a little Java class, throw it in, and then you'll get plots. It'll actually make plots for you of the trinucleotide context. Any other questions? Does this make sense where we're going? Okay, so let me, so I'm going to give a little bit about the mathematics and how this works. It's actually very simple. And then I'm going to show you lots of data and try to explain how many sequencing vendors do not do a very good job of estimating these quality scores. And then we'll show you command lines and things. Okay, so the principles. So how it's going to work is all we're doing is we're counting up the covariation of error with all these various parameters. So I'm listing here the original reported quality score. There's the machine cycle of where the base was read. I showed you the dinucleotide context. So all you're doing is you're tabulating when you saw these covariates of error, how many times did you see that, that combination of things, and how many times was it an error? So maybe I saw Q17AA 1,000,000 times, and maybe it mismatched the reference 1,000 times. And so you can do one divided by the other, and you have a quality score, right? Very simple. And so all it's doing is a piecewise tabular correction where you make an independence assumption for all these various covariates, and you have a correction factor based on the quality score, a correction factor based on the machine cycle, and another one based on the sequencing context. It's very simple, but surprisingly very powerful. And so the way you decide if something is an error is simply by looking at the read and does it mismatch the reference or not. So sequencing errors will mismatch the reference, non-sequencing errors will match the reference, and then you may wonder, what about real genetic variation? So real genetic variation is not error, and it also mismatches the reference. And so what we do is we simply discount all the sites of known genetic variation. And when you do this, because dbSNP is so massive and so great, 99% of the real variation in your sample is found in dbSNP. And so you're sort of discounting those sites, right? So now everything that mismatches the reference is counted is assumed to be an error, and the things that don't are assumed to be real. We'd have this very simple smoothing procedure where you add 1 to the number of observations and add 1 to the number of mismatches, and you turn that into a quality score. So this will be, there's 10,000 reference mismatches over 1,000,000 times you saw that combination, and that's a Q-score of whatever it is. Make sense? Okay. And then Eric wanted me to point out that it's important to do indel realignment first here, of course, because as you saw, you were sort of cleaning up all these mismatches around indels, right? And so those mismatches would be counted as errors in the read. And so by cleaning those up, you're no longer penalizing the reads, or no longer penalizing the bases. So that's why it's important to do indel realignment before this base quality score recalibration procedure.
So is this very much dependent on the DB?
I'm sorry, I really can't hear you at all.
This is dependent on the dbSNP?
Right, so it uses dbSNP to discount these.
That is not in dbSNP, then we can't.
Right, so it's interesting. There's actually a lot you can do. So the question is about, you require this dbSNP. What if I'm sequencing in these crazy organisms that don't have dbSNP? What can I do? So we might as well talk about it. There's lots of things you can do. It's very interesting. All you need to do is discount the sites of probable genetic variation, right? So there's lots of strategies. One is you could make an initial round of SNP calling. You could take your data, make an initial round of SNP calling, and just say, I'll take all the SNPs where I'm 99% confident, I'll use those as my database of genetic variation. Because all you want to do is discount the sites that are probably genetically variant, right? So it's very fuzzy because what we're trying to do is just get rid of these crazy systematic biases. We're not trying to be incredibly accurate, we're just trying to get rid of the crazy biases, right? So if you're sort of off by a little bit, it's not a big deal. And I'll show you some examples. What's up in the back? Wow, this room sucks. I can't hear you at all. It's a big vacuum.
Is this better?
A little bit.
Do you account for interactions among the covariates?
Okay, so no, it doesn't at all. So the question is, you make this independence assumption, is that a problem? So what we have here is the covariates that we're using, we've done lots and lots of testing to convince ourselves that it's mostly independent, so it's not a big deal. So if you were to throw any old thing in there, it's probably not a good idea if there's covariation, because it doesn't account for the covariation among the covariates. But for the ones we're using, original quality score, machine cycle, dinucleotide context, these things are pretty much independent, so it doesn't matter. It's a good question. So if you were to throw in like whether the machine operator had a cold that day, or whether they sneezed on the lane, then that would probably be independent of the quality score.
I wonder if, whoa, that's loud. I wonder if you could, if people do put in some kind of control DNA to assess the exact, parameters for the error on in a specific lane, just a 1 or 2%. How much control DNA would you need to do something?
So I have read a bit about people doing these sorts of things. So you may know about this project called the Genomes in a Bottle at NIST. It's trying to do this sort of thing. I don't know any specifics though. Well, the problem is so a quality score of Q40, right? In order to be able to differentiate between a Q40 and a Q39, you're going to need thousands and thousands of observations, right? Maybe 100,000 observations in order to differentiate between those quality bins. And so it's really a matter of how accurate you want to be, right? If you want to be accurate within a few Q units, then you need 10,000, or you need 10,000 observations of every combination of covariates. But if you want to be more accurate than that, you need much more. Does that make sense where I'm going? So it really depends on what your goal is and like how accurate you really need to be.
Do this on whole exome, or is it limited?
An exome is perfectly fine, yeah. So you need, I think our baseline is you need something like 15 million observations, so 15 million sequence of bases, which you have way more than that in an exome, so that's fine. If you have, for example, if you're doing a very small target region and you only have, for example, 100,000 bases sequenced, it's probably not a good idea to use this procedure, right? Because you just can't accurately estimate those quality scores, right? But anything on the whole genome or whole exome scale, perfectly wonderful. Any other questions? Okay, so let me show you some data, I think. Yes, so this is my most favorite slide I've ever created. So what I'm showing you here is some pink dots and blue dots. Pink dots are the original quality scores as they come out of the machine, and the blue dots are after you've run through this base quality score recalibration procedure. And I'm showing you for a variety of sequencing technologies. And so what makes this slide great is it shows that I'm not biased by any sequencing company because they're all pretty terrible. And so what you see here is that, for example, we'll take this one. So on aggregate, as you say that the bases are Q23 or whatever, they're in fact, when you look at them in aggregate, they're in fact Q17, right? And so you may think a difference of 5 quality score units is not a big deal, but unfortunately it is a big deal because, of course, the Fred scaling procedure it's a logarithmic sort of scale. So 5 orders of magnitude difference in the quality units is actually an incredible difference in evidence, right, towards mutation or not mutation. So being off by a factor of 5 Q units is actually quite a big deal. And then moreover, perhaps there are other sequencing vendors that try to do a better job when you look at it in this view, but through whatever they've done to make the quality scores accurate, they've added these crazy systematic biases. So for example, 454, they have very long reads, and perhaps the quality scores are more accurate, but they have crazy biases along the length of the read, right? Where the beginnings and ends of the reads are grossly overestimating or underestimating the quality scores, when in the middle of the reads, it's fine, right? So this would create crazy effects when you try to call mutations. So everyone see why that's bad? Okay, and then you may think, okay, these are really old technologies. What about very new technologies? So maybe this one or this one. And you can see that it's still quite terrible. And in fact, these people have added something to their quality score estimation procedure to create these crazy 5-cycle periodicity bias in their quality scores, right? So maybe you can't see it quite so well, but there's these dots, they go up and then they reset and go up again. That's why they're sort of very wide here. So what's happening is that every 5 bases, they reset their Gaussian mixture model or whatever they use. And so they've created this bias in their quality score estimates as a function of machine cycle. Does that make sense? What's up?
Have you looked at any ion or biodata?
Oh, yeah, I got a pack bias slide coming up. Everyone always asks me. It doesn't matter. The problem is no one understands quality scores. So these companies, they just don't understand how important these quality scores are. And so they don't do a good job of estimating these things. If they wanted to estimate them very accurately, they could. They just don't want to, or for whatever reason. And so they're all bad. There's not a sequencing vendor out there that's good yet. Hopefully one day they will someday be good. But the thing is, all you have to do is use this program, and these biases go completely away, right? So the blue dots are perfectly on the lines, on the line, on the line. All these biases are completely removed by using the GATK. So one final thing to say is that, so I've shown you quality score accuracy, the bias by machine cycle, and also the bias by dinucleotide context. And they're all quite terrible in terms of sequencing context. Good? Is everyone convinced that they should use the GATK to have accurate quality scores? What's up?
So you compare to the reference sequence. In the reference sequence also sequenced based on one specific technology. Does this affect the results that you got? For example, maybe there is some systematic error in that reference sequence too?
Yeah. I see what you're saying. But the problem is, I mean, the reference is, I mean, it's quite different, right? It's like this consensus that's built by this highly accurate Sanger whatever assembly. And so, I mean, I suppose that there is some systematic bias there that you're picking up here, but I don't think it would account for these crazy differences in like read length and things, right? Like maybe there's a bias by sequencing dinucleotide context, but by machine cycle, it seems a little odd, right?
Ryan, systematic bias is like caught by being snippet.
Sorry, what?
Systematic bias is like caught by being snippet. Right? So if you have differences in the reference,
Yeah.
they're shown as SNP, they appear as variation over a few minutes, high frequency variation. They're captured as SNP, so they can filter out here.
Sure. So things that are identified as variation, like genetic variation in the population, that stuff would be discounted in this procedure because you're skipping all DB SNP sites, for example.
Yeah, but maybe the difference is based primarily in the same sequence.
It doesn't matter, right? The only issue is are there errors? Are there mismatches in the reference? If the reference is wrong, every sample is a mismatch.
No, I'm saying that the BSNP was also based, for example, Sanger, which has exactly the same systematic equivalence. Then you're not going to find a mismatch. I'm not saying. There is just another thing that I remember one of the retreats talks that we had, they say that some of the samples, I think the library size can affect the sample in such a way that actually a mutation happens after the library was prepared.
Mm-hmm.
So actually the sequence was correct. It's the library itself that changed. It's just something to take into account.
Yeah, but even then, so you would want to, so those things would show up as errors, right? And you would want to discount.
It's going to show as mismatches, but actually was not a sequencing error.
Yeah. I see what you're saying. So it's not a sequencing error.
I don't think it's going to affect it. I'm just saying as a sake of curiosity.
Okay. Yeah, I mean, it's interesting because everything here is very fuzzy, right? But the point is, you're trying to get rid of these like very, very gross systematic biases that if you're off by a little bit or if there's different kinds of issues, it really doesn't matter because you're trying to correct for like crazy things like this, right? So anyway.
So quick question. So the last row, okay, that's the plot for where you look at the base before that, right? So what if you plotted the base after that?
Mm.
Do you see the bias or just
So I would say that you would probably not, and the reason why is because these biases are a function of the machine like reading. So the machine will read A followed by another A, and then somehow get confused and overinflate its estimate in the quality score, right? So if you were to look at A, so if you look at the first A followed by another A, the machine hasn't read, as long as you're doing it in the order that the machine read the dinucleotides, you should see the bias in the second base. Does that make sense?
For the next base, that determines the binding strength, right? Something, okay, next strength, COC, and then INP. The binding strengths are different, and then the base before that would be positive, right?
Right. So it depends on what the sequencing machine is doing, right?
Yeah.
So if the machine would get confused by that, then you would see the bias. Otherwise, you wouldn't, right?
Yeah.
So this is a property of the sequencing itself.
Yeah, it would be good for you to show, okay, the next show that, instead of show that, to look at the base before that, it would be good to show the base after that.
Sure. Sure. It's an easy thing to investigate, actually.
Yeah.
Any other questions?
Just another question.
Oh, what's up, man?
You should have the opportunity to just check for, it's not exactly related to your quality, but check for other systematic errors besides the ones that you already know when you compare all those sequencing technologies.
I'm not sure I understand the question. Sorry. I just can't hear anything.
For example, that 4x4 has a huge tendency of creating homopolymers because
Yeah.
were you able to detect anything else besides those?
Oh, I see. I see. So we've tried lots of things, actually. We've tried even crazy things like time of day and just crazy things. And these are always the ones that we come back to. They seem to be very consistently show a bias. We've done things like homopolymer length. We're experimenting right now with things like repeat length to try to capture things about short tandem repeats. And these things are interesting, but they haven't proven to be so successful yet. So I would say these are the big ones. We haven't found others.
Yeah, we know that there are many factors that cause the bias according to sport. How can you recalibrate them simultaneously, or you just recalibrate it one by one, for example, cycles, anything that you can have?
Right. So the way this works is it makes an independence assumption. So you have a correction factor based on the machine cycle, and another one based on the sequencing context. And so you do those corrections independently. And so what you're seeing here is that when you make that assumption, when you make that independence assumption, you're not adding a systematic, you're not like having a bias by not accounting for the covariation. Otherwise, these blue dots wouldn't be on the line here, right? Or rather, another way to say it is that what the little variation that you do see in the blue dots is due to not accounting for that covariation. And so you can see it's very, very small.
As this step could also be applied for IC data.
Yeah. I mean, as long as you have enough observations, yes. And you would want to discount the sites of known variation. But yeah, as long as you have enough observations, it would still work, yeah.
Just to interrupt one moment, if you're asking a question, try to come to the mic. I think it's going to be, because we have a lot of questions, and they're not recorded, so it's going to look really weird on the video. And if Ryan has to repeat every time, that might be a little tedious for him. So maybe try to come to the mic if you have questions, or Ryan, if you can repeat them.
How are we on time?
I think we're doing wonderful.
Okay, good. So let me continue a little bit more. Let me try to remember what's next. Oh, yes. Okay, so I wanted to show you, we talked about PacBio, right? I promised you a PacBio slide. And so what's interesting about the BQSR, and so this what I'm talking about now is new in the GATK 2.0 or the GATK full or whatever you want to call it. So what we saw was that you can use this exact same procedure that we use for base mismatches, and you can do the exact same thing for indels. And you can create an estimate of the base insertion error rate and also the base deletion error rate. So right along the string of quality scores that get spit out by the machine, you can also have base deletion quality scores and base insertion quality scores. So now every read has 3 strings of quality scores, and when you do that, you can get incredibly accurate indel calls. So what I'm showing you here is for a HiSeq machine and a Pacific Biosciences machine, the estimate of the indel quality as a function of a sequencing context. So this is now a 9-base sequencing context. No, 8-base sequencing context, where you start with 5 A's and you end with these 3 bases here, right? So you sequenced AAAA, followed by ATC, and when you do that, you have an estimate for the indel error quality, right? And so some interesting things pop out from the slide. The first, you see that there's a crazy different profile between a HiSeq machine and a PacBio machine, right? So the first thing is that the HiSeq machine has a much higher quality indel error rate, so it's a very low indel error rate because the indels are high quality, right? So any indel in a read is correct at a rate of Q40, right? So 99.999. That rate of correctness. But it has a crazy bias in terms of sequencing context, right? So where there's 5 A's followed by 3 more A's, this is kind of crazy. So when you have 5 A's followed by 3 more A's, it's correct at a rate that's much lower, right? So it's biased by its sequencing context. And furthermore, I'm showing you 8 different read groups, so 8 different runs of the HiSeq machine. And you can see they're all over the place, right? So that's what these different shades of green are. They're sort of, one run it was correct at a Q40 rate, but another run it was correct at a Q35 rate. So all you did differently was you ran the machine a second time on the exact same sample, and it was correct at a very different rate, right? Okay. So contrast that with PacBio. PacBio makes way more indel markers in their reads. So many that most of them are in fact wrong. They're correct at a rate of Q10, very low. But what's great about it is that it's completely unbiased. It's perfect. It's beautifully unbiased by sequencing context, right? So you don't get into these. You don't have these systematic problems where, you'll find mutation calls in a certain context, and you're not sure whether they're real or not. Whether there's this bias based on context. Does everyone understand the contrast? Okay. And so this is sort of the new feature in sort of the BQSR in the last, I don't know, 6 months or something, is that we, in addition to estimating a base substitution error rate, we have a base insertion and a base deletion error rate. Okay, and so now I'm showing you for the exact same thing for HiSeq. So now these are the same sorts of plots you've been looking at, but I have base substitution here, I have base insertion, and base deletion. And so you can see that in addition to sequencing context issues that I showed you, there's also an issue in the accuracy of the indels as a function of the machine cycle, right? So at the beginning of reads, you overestimate the quality score, and at the ends of reads, you underestimate the quality score. It's very wild. And you can correct for that using the GATK. Is everyone clear? Okay, so finally, let's go through some command lines. And so as most of the GATK tools that you're going to see, it's a 2-part thing, where the first step is you create a model, or you create something, and the second step is to apply that thing. So what we're going to do is we're going to create this model of the errors based on these covariates, and then we're going to have this massive table, and we're going to apply that table to the data, writing a new BAM file with exquisitely accurate quality scores. Okay? And so let's go through this. So you sort of read these workflows from top to bottom. So it starts here, where we have the original BAM file and the database of known genetic variation. And we're going to combine this together using this procedure called base recalibrator. And this is what the command line looks like. So you have Java, the jar, and this is the name of the walker. It's called base recalibrator. You have the reference, the input BAM file, your genetic variation. So we use dbSNP for mostly the SNPs, and we have another database of high-quality indels that we use. The output here is going to be a GATK report file. So this is the GRP, GATK report. And then you create all these beautiful plots come out of the procedure in a PDF. So this is what your command line looks like. And when you do that, you'll get the GRP file, the GATK report, and also some plots. And so what you can do with that is you take the same command line, run the procedure again to get sort of the before and after look. So you'll get the pink dots and the blue dots on the one plot, so you can sort of compare before and after. So it looks like this. It's the exact same command line. All you're doing now is you're passing in that report. You had the report from the first command. Now you're passing it in. And so you get post-recalibration plots. So now you'll get pink and blue dots on top of each other, and it shows you how it works. OK? And then finally, to create the actual recalibrated analysis-worthy BAM file, you would take your original BAM file, take your GATK report file, put them together using a walker called print reads, and you get a recalibrated BAM file. So it looks like this. You say print reads. You give it your recalibrated report file, and outputs the recalibrated BAM file. Very simple. So now you have new quality scores. You retain the original quality scores in this tag called OQ, just in case you're a little nervous about losing your quality scores. And that's that. So if you're doing the base substitution, base insertion, and base deletion, all of your reads will now have 3 strings of quality scores, and that's that. And how do you know if it works? Well, you can just look at these plots, pink dots on top of blue dots, and if it's working well, the blue dots should be perfectly on the line, right? Because we were saying before, if for some reason this independence assumption is not so great, what you'll see is that there'll be a systematic bias, right? Like maybe you haven't corrected for all of the variation in the data. And so you'll see, so maybe you'll see that in the blue dots. So that's just a way to sort of QC your results and make sure everything's working well. And that's basically it. And there's some further reading. OK, what's up, man?
So print reads also generate those nice graphs? Print reads?
No. So the way it works is the output of base recalibrator, actually, that's making the plots. So it creates this report, right? This table. I saw Q17 and it mismatched the reference this many times, or I saw cycle 10 and it mismatched the reference this many times. And then all you're doing to make the plots is just plotting those tables. It's very simple in R. You read the table and plot it. It's like the easiest thing in the world. So that's why this creates the plots. The reason why you have to do it again is so that you can get the before and after, right? I guess technically you could create a new BAM file and then rerun it again, and you could do the same thing. But this is sort of a way to do it without creating a new BAM file. Right, you pass it in the original table, it creates a new table, and then it can plot both of them on top of each other. It's just a shorthand, so you don't have to, because these BAM files are very large. And so writing them out again takes a long time. OK, anything else? OK, that's it then.